DNA topology describes the three-dimensional arrangement and spatial relationships of the DNA double helix beyond its primary sequence and secondary structure, which is the double helix. A key aspect of DNA topology is supercoiling, the coiling of the DNA helix upon itself. This is essential for packing meters of DNA into the microscopic cell nucleus and for regulating critical cellular processes. DNA naturally exists in a relaxed state when its ends are free to rotate. However, in the cell, DNA is typically circular, as in bacteria and plasmids, or anchored at points along its length, as in eukaryotic chromosomes. When the DNA helix is underwound, meaning it has fewer helical turns, or overwound, meaning it has more helical turns, relative to this relaxed state, torsional stress builds. The DNA relieves this stress by twisting into loops or coils, forming supercoils. Supercoiling is quantitatively described by the linking number, abbreviated LK, which is the number of times one DNA strand crosses the other in space. The linking number difference, denoted deltalk, compares the actual LK to the LK of relaxed DNA, called LK0. The formula is deltalk equals LK minus LK0. Supercoils can be negative or positive. Negative supercoiling, where deltalk is less than 0, results from underwinding the DNA helix. This is the predominant form in most cells. It reduces the number of helical turns, making it easier to separate the DNA strands. This strand separation is crucial for processes requiring DNA melting, such as transcription initiation and DNA replication. Positive supercoiling, where deltalk is greater than 0, results from overwinding the helix. This compacts DNA, but makes strand separation difficult. It often builds up ahead of replication forks or transcription complexes as they unwind the helix, acting as a barrier that must be resolved. Cells precisely regulate supercoiling using enzymes called topoisomerases. Type I topoisomerases make transient single-strand breaks in DNA, allow controlled rotation or strand passage to change the linking number by 1, and then reseal the break. They primarily relax supercoils. Type Roman II topoisomerases, such as DNA gyrase and topoisomerase Roman IV, make transient double-strand breaks, pass another segment of DNA through the break, and reseal it. They can introduce negative supercoils, as gyrase does in bacteria, or relax both positive and negative supercoils. They change the linking number by 2. In eukaryotes, supercoiling is intimately linked with chromatin organization. Negative supercoiling facilitates the wrapping of DNA around histone octamers to form nucleosomes. The assembly of nucleosomes and higher-order chromatin structures also constrains DNA supercoils, creating localized regions of negative supercoiling that influence gene accessibility and activity. This video is part of our free DNA topology and supercoiling course for biology students, with over 100 subtopics to explore. Find the link in the description.